Welcome to the latest edition of the Mind Gut Conversation podcast, a place to learn about new ideas from thought leaders in the area of optimal health, the science of mind-gut microbiome interactions, food, and the environment. Today, I have the great pleasure to speak to Leo Grady, AI expert and CEO of the microbiome company Jona. Leo got his Ph.D. in cognitive and neural systems from Boston University and is the founder and CEO of Jonah and CEO in residence with Breyer Capital. He's internationally recognized for his work to deliver AI in healthcare for 20 years at pioneering Bay Area startups, multinational medical companies, including Siemens, and most recently as CEO of Paige.ai. In this role, he led the company to become an industry leader, internationally launching groundbreaking products and receiving the first ever FDA approval for an AI product in pathology. He authored 2 books on AI, over 100 peer-reviewed scientific papers, and is an inventor on over 300 patents. Winner of the Edison Patent Award, he was inducted as fellow in the American Institute for Medical and Biological Engineering. As founder and CEO of Jonah, he has developed a stool microbiome analysis platform which not only uses state-of-the-art analysis approaches to study the structure and function of the gut microbiome from individual stool samples, but also provides users with a unique AI-assisted interpretation of the results. Welcome, Leo, to this podcast. I'm really looking forward to hear about your experience and your venture into the microbiome space using a different approach than most other people have done before. And so, let me start out with this question. You have a career that's deeply steeped in artificial intelligence research and development. So, how did somebody like you get involved in the gut microbiome?
Yeah, it's a great question. I actually grew up around microbiology. My father was a virologist. He ran virology for New York State Health Department. And so I grew up going to the lab with him and talking to him about viruses and bacteria. And so that was something that was just a part of my childhood and growing up. And then as I went into adulthood, a number of family members ended up being diagnosed with Crohn's disease, colitis, celiac. And for each of them, it was a journey of really trying to find a diagnosis that in some cases took many years. And as you know, the microbiome is implicated in etiology for each one of those different diseases. And then at the same time, over the last 10 years, there's all this incredible science coming out showing the microbiome linked not just to GI disorders, but autoimmune conditions, to behavior, to oncology, to drug efficacy, to liver disease, to obesity, even mental health, depression, anxiety, Parkinson's, Alzheimer's, all being linked to the microbiome. And I kept waiting for all of that science to get translated into the clinical world, but it kept not happening. And I felt that one of the barriers was the complexity of the microbiome and a need to really build up sufficiently large data sets to really understand what's going on, to model it computationally, to understand it computationally. And then that would really provide the basis to clinically translate a lot of this work. And so ultimately, it's an area that I've been excited about for a long time, following for a long time. I feel like it's personally touched me. And now it just seemed like the right time where we do know enough. The AI technology is an interesting place. Sequencing is cheap enough that we can start to do this at scale. And that's why I jumped in.
So you formed this company, Jonah, which offers several services. And I'm just going to go over these: clinical grade, deeper sequencing, AI-backed, actionable, and personalized recommendations, contextual understanding, and continuous learning and updates. So let me start with the first one. So clinical grade, deeper sequencing. What do you mean by that?
We do stool testing. We do gut microbiome. It's a stool test that you can take at home. We ship a kit to your house, and you ship it back to the lab. We do deep shotgun metagenomic sequencing. identify all the bacteria, the viruses, the fungus, the protists, the archaea, everything that's in your microbiome. And because we're doing that with sequencing and not PCR, we really identify what's there. We also identify the genes as well as the species. And because of the depth of sequencing, we can get down to a strain level.
And so you mentioned the genes. So can you also make any conclusions about the function? I mean, obviously there's a shift within microbiome science in the last few years, kind of away. I mean, not totally away, but incorporating more and more the functional aspect, not just who is there, but what are they doing?
Yeah, absolutely. And that is a really important element of today and of the future. I would say that at the moment, there's more literature on linking different species of who's there, as you say, to different disease processes. Um, we are learning, as you point out, more and more about function and the genomic signatures for that. I'll also say a few other things is that the genomic analysis allows us to do a few other elements as well. One is understanding antimicrobial resistance, whether certain species in the person's gut are going to be resistant to certain antibiotics. And the other thing is that there's some new research that's actually starting to show that rather than looking at the association of different species with different disease or disease processes, actually just looking at the association of different genomic components in these species with different diseases. And so it's a bit of a shift, not only toward function, but toward looking at the DNA of these organisms and linking that to disease processes rather than the species as a whole. And so I think that's an exciting area of research. And as that literature grows, having the genetics will allow us to leverage that as well in terms of trying to analyze somebody's microbiome.
Yeah, I mean, there's obviously, often cited example, there's these different strains of E. coli, some being beneficial, others like enterotoxigenic, E. coli being, really dangerous. Um, and that probably applies to a lot of the species that have sort of been, reported by these tests, um, like even Akkermansia or um, Prevotella is a good example where it spans the whole spectrum. Um, so with your test, you take this into consideration. You don't just say there's too much, because like when I see, in my clinic, I mean, I see patients coming in with these 20-page reports, and it lists the good and the bad ones, and patients are generally totally confused. The only thing that they and maybe that's intentional. The only thing that they get out of these reports is to know which supplement is being recommended for their particular microbiome. So if I do the test with you, I haven't done it yet, but I've ordered the test, so I'm looking forward to it. Um, well, do you go down into these, as I said earlier, into the strain level and in terms of the recommendations, not just based on if one species is there or highly abundant, then you said it's a good thing or it's a bad thing. So I mean, does your test and program take this into consideration?
Yeah. So I think this paradigm of good guys and bad guys is one that is very simple. And certainly within the probiotics industry, it's very easy to say, take our supplement because we've got all the good guys, or take our drink or whatever it is because it contains good guys, and take antibiotics for the bad guys. The reality is a lot more complex than that. It's the entire ecology that matters. Certain organisms being too low can be problematic. The same organisms being too high can also be problematic. Martin Blazer has, all these stories about H. pylori and how it's generally vilified, rightly, and a bad guy, quote unquote. But H. pylori, the absence of H. pylori, is also associated with things like GERD. And so it's not so simplistic and easy to say that H. pylori is either strictly good or strictly bad. I think there they're elements of both, and we can turn the dial one way or the other. And that's true of not just H. pylori. I mean, these organisms have co-evolved with us and have been around for a long time. And so part of the change I think we need to make in the whole field is to get away from this paradigm of good and bad. But to your question, what we do at Jonah is basically when we do this shotgun metagenomic sequencing, which identifies the bacteria, the fungus, the virus, everything down to a strain level. Then what we do is we built a large language model, a form of AI that reads the entire microbiome literature. Every single month, there are more than 2,000 papers published in PubMed on the microbiome. So nobody can read all of these. Doesn't matter how many CME credits you get, you're not going to be able to get that entire literature. And so the AI reads all of that and then provides a summary for the individual of all of the diseases, all the conditions, all the symptoms, the allergies, everything that's been linked to a microbiome configuration that we find in that individual. And for every linkage that we describe, the AI cites the relevant papers. We link out to those papers in PubMed if you want to go read them yourself. We tell you how much evidence there is, whether it's preliminary or more established. And then insofar as there's some issue found in your microbiome, then the AI goes through the literature and sees what you can do about it in terms of diet, in terms of lifestyle, or even in terms of medication, to be able to say, these actions that you can take would move your microbiome in a better direction, or these actions have been shown to be more efficacious for somebody with one type of microbiome versus another.
Yeah, I mean, that's really a huge, I mean, this is really the point that has attracted me the most to, to your program, that you do these additional services, not just the sequencing, but these additional services that normally a patient either goes to their physician, the great majority of whom do not know the answers because they're not trained in microbiome science, and whatever they say may just come from some marketing from some supplement company. So to give this information, put it into context, and then provide some actionable recommendation, I think that's one of the biggest strengths of this program.
Well, I want to elaborate on the point that you're making there, because I really think that separating out the lab testing from the AI, from the interpretation, is one of the key opportunities with AI. And if you think about what I did personally in my own career, building AI in pathology, building AI in cardiology, building AI in radiology, it was a very similar paradigm, right? So just prior to Jonah, I was CEO of a company called Paige.ai that built AI systems in pathology. And so we took digital pathology images. We had the first FDA-approved AI system in pathology to detect prostate cancer on needle core biopsies. And what we did there is we didn't run the lab. It didn't matter who did the sequence or who did the tissue extraction or the cutting or the staining or the mounting or the scanning. And it didn't matter what scanner you used. But at the end of the day, our AI could look at that digital image and find the cancer. And so from a healthcare standpoint, there's a real separation between that lab work and the AI interpretation. And so with Jonah and with the microbiome, I see we're taking a very similar approach. So if you're doing the sequencing, we partner with a lab that can do the sequencing, but even if someone's doing the sequencing themselves, we are an AI company and a software company that can interpret that data regardless of who did the sequencing. So long as it's of sufficient quality and depth that the data is good, the AI can provide that additional service and really create, I think, more value out of the data than just a big long list of species.
So does your algorithms, do they take into consideration the quality of the publications? I mean, there's obviously a lot of stuff being published that does not meet that criteria. So your algorithms take this into account and do not include texts or publications that do not meet those criteria.
Yeah, we take into account quality at two levels. So first, there's just an inclusion bar. So we only include peer-reviewed papers, only papers that were studying humans, only stool samples, only sequencing, had to be in vivo studies, not in vitro studies. And they had to be studies that were uh, in patients of age 5 or older. And so all of these different inclusion criteria says we don't even look at papers that are not peer reviewed or don't fall in, animal studies that don't fall into those criteria. But then even papers that do meet those criteria, we give them a quality score, which is a function of the size of the cohort of the study and the impact factor of the journal that they were published in. We don't want to be in a position of trying to assess everybody's methodology or judge their science. We really wanted an approach that's standardized where we can leverage just cohort size and the impact factor of the journal. And so with that, when we do the summary for somebody, we weight studies differently depending on that quality score. So like a large study in the New England Journal is going to get more weight than a smaller study in a second tier or third tier journal.
So I'm not an expert in AI, even though I'm fascinated by it. So how much biological or medical or scientific supervision or input does this have? So is there somebody, like, for example, with the recommendations, is there somebody with knowledge in the field that would look at the results, or do you trust 100% on the output from the AI?
So we do have an amazing clinical advisory board. We do have some MDs on staff. With the recommendations, though, remember, we're not practicing medicine, right? What we're doing is saying these papers have shown that with this microbiome configuration that you have, a FODMAP diet is more likely to be successful or less likely to be successful. And that there's, this was a high quality study or a lower quality study. So we're not managing these patients. We're just providing this information to them and their physician to say, this is what the study says, and this is the quality of the study. Ultimately, we're just providing them that information and not trying to practice medicine in any way.
Is the quality of these results high enough? I'm just thinking about, your main target are patients, obviously.
Well,
But
doctors are a really important constituent too, for sure.
yeah, but I mean, this kind of product could equally be useful for even scientists that work in the area. Like if you write a review article on microbiome and IBS, you could use that as well. I mean, that information.
Yeah, it's funny that you say that because it's pretty frequent now that people will reach out to me, our partners or people that we know, and say, hey, what's known about NASH in the microbiome? What organisms are linked with metabolic disease in the microbiome or whatever it is? And so these are things that we can just pull out of our knowledge base now that the AI has read all of the papers in the literature. And we can really highlight, which species are associated, what's known about those species, et cetera, et cetera. So from a company standpoint, we haven't really built a dedicated research tool or product. It's certainly something that we plan on doing, probably in conjunction with the life sciences industry and the biopharma industry, to really help them better understand stool samples from trials and whatnot. But we're certainly open to working with the academic community on this as well, because ultimately we want to advance microbiome science, and we feel that it's more than just we can do on our own.
Yeah, I mean, the prospects or the potential of this is really amazing. When I think about this, I mean, just having had this personal experience, for our newsletter posts, which are written by, very smart students and people who have just graduated, and then I'm going over each of those. So just to find out, what we would get if we had AI do the same article, and it is actually pretty remarkable, how, I mean, first, I mean, in terms of the style and the composition of the article, probably AI beats most people that I work with. And but the content is usually correct, and it's very careful. It doesn't make these claims, it points out where can't really make this conclusion, or this is ongoing research, which I like. I mean, that's kind of what a system like this should do. Is I mean, do you feel the same way that if you have, I don't know if you've done this, but to show data sets to microbiome expert and say, can you comment on our findings, and then compare this to what the AI interpretation is and recommendation is that, do you have a similar experience?
We do, and we work with our scientific advisory board includes some of the world's top microbiome scientists, and we're frequently looking at results with them, and they're extremely knowledgeable about the microbiome and different organisms. At the same time, they're human. They can't read 2,000 papers a month, and even if they could, and even if there were 300 papers on Parkinson's disease and the microbiome, trying to remember what each of those 300 papers said and then apply that knowledge to look at somebody's microbiome is something that's very difficult for humans to do. And so they may remember, okay, F. prausnitzii is low or Akkermansia is low. That's a bad thing, and it's linked with a few different diseases. But to your point earlier, the whole ecosystem matters, and it's not just Akkermansia being low; it's Akkermansia being low and something else being high, and then the configuration of a number of these organisms. And I think that's very hard for people to do mentally. But this is where AI can really help us by being systematic and reading the literature and then applying these papers to interpret the person's microbiome. We can be really consistent and then also always cite back to the literature so somebody can go read it themselves and come to their own conclusions if they want to.
So you emphasized earlier that you only look at human studies, or have AI just look at human studies. One unique thing about this field is, as it is in many areas of science and medicine, that this big translational gap between,
Right.
and it's particularly strong, I think, in microbiome research, that all sorts of claims are being made, and started really with the brain gut microbiome interactions, that all of a sudden, articles in the lay press came out that depression is caused by dysbiosis and Alzheimer's, and that's usually based on mouse studies. And
Right.
then when you look at the actual translatability, I always say it's lost in translation. 90%, you cannot see human studies that really confirm this unequivocally. There's a few exceptions. So by not looking at the preclinical literature, is that are you concerned about this, or that you don't take this into account? Like for example, the whole thing about, which I'm most familiar with, anxiety, depression, Parkinson's, Alzheimer's, those areas.
It's a tough question, right? Throughout medicine, we face this same issue, right? I mean, I've heard people in oncology say it's great to be a rich mouse because we can cure any cancer in a mouse. But those cures don't always translate to humans. And it's never clear when they're going to translate and when they're not going to translate. And even though we may have a good model for different diseases, I think animal studies can be hypothesis generating, and I think they can allow us to work out certain mechanisms. But if what we're trying to do is look at a human person's microbiome and draw any kind of conclusion from that and link that to depression or anxiety or Parkinson's or whatever it is, I don't feel comfortable leveraging the preclinical studies there. So it's not that we can't read them. It's not that we couldn't incorporate them, and other people may have different views. one of the great things about where we are now with microbiome science is that there are enough human studies that we can start to draw some conclusions. And I would say that we don't need to necessarily go to the mouse models. But I think one could argue it either way. I would just personally feel more comfortable and more solid being able to say something about a study in human being more relatable to analyzing a human's microbiome.
Yeah, and I would totally agree with that statement. I mean, the complex, and particularly if you deal with the brain, I mean, the complexity of the human brain is just, I mean, it's like two different machines. Totally, there's not that much. There's some similarity on the most basic level, like on the amygdala level, but everything above it is totally different. So, to make these claims, what you see in the mouse necessarily has to relate to a human pathology is just not. Unfortunately, that mistake is made, repeatedly. And I mean, a big amplifier of these mistakes is really the lay press. Like something comes out from a mouse study in Nature or in Science, and immediately the heading is, do we have a solution to human depression? Or something like
Or
that.
autism or things like this.
Autism is another good example. Yeah.
Yeah, and before I started the company, this was one of my motivations because I would, dutifully take probiotics and buy them. And people, sometimes even doctors would say, "Oh, well, those are the high quality probiotics, those are the low quality." And I'd look at these different species levels and I would say, why these species and why not these species? And why are these the good ones and these are the bad ones? And what problem am I trying to fix? And why is this relevant to me? And I was reading, the lay press, I was reading the scientific literature, and there were all of these amazing findings, some of them in animals, some of them in humans. And it just seemed like there was a big gap in trying to connect that science and that research to understanding of my own microbiome or of what any particular probiotic might do. And so, one thing that we do or we don't do at Jonah is create probiotics or sell them or market them. Not that we're against it or anything like that. It's just our view is that there's so much information in the microbiome, and we want to really be loyal and true to the patient and the doctor to provide them that information and not use it as a mechanism to sell some supplement. So it's partly from the standpoint of our values, we haven't gone in that direction.
But in terms of endorsement, I've seen some of that on your website. So you do endorse certain certain probiotics for certain. I do not.
We don't endorse any particular brands or anything like that. What we can say sometimes is, so let's say somebody's microbiome is associated with a certain condition or a certain disease, and that their microbiome is low in certain organisms that you can buy in commercially available probiotics. So we may say, find a probiotic containing these organisms that may help reduce that association. Again, we don't know causality there, but that might reduce that association. But we don't say buy brand X or we'll sell it to you or anything like that.
I mean, this would obviously be something impossible for AI to decide because there's so few human studies with probiotics that would meet, for example, FDA criteria for approval of a drug. I've seen it many times that the companies that have a successful well-selling probiotic, the last thing they want to do is do a big controlled randomized study because the chances are that it comes out negative. So how does AI evaluate really the paucity of those kind of studies? I mean, there's a few exceptions now. There's a few companies that are doing this or are in the process of doing it, which I really admire. And actually, this is one of my criteria. If I go on the external advisory board of a company, if they are willing and have a track record of doing this. But how does AI evaluate this? That so many claims are based on really either in vitro studies on cell cultures, and you can show anything on cell cultures or on, mouse intestines. So,
Well, if there are probiotic studies that meet our criteria of human and sequencing, et cetera, then it's something that we would include in the AI, and we would assign a quality score based on the size of the cohort and the quality of the journal. So we would treat it like any other study. One thing that I personally struggle with when I look at that literature is a lot of these studies are done using, quote, probiotics, but the probiotics aren't selected for specific strains or particular species. And there seems to be a view that all probiotics are the same. And if we know that certain microbiome configurations are associated with different diseases, or that your particular deficiencies may be associated with particular diseases, it would seem like we would want to control for inserting the right species, the right strains, and do the studies in that way. But again, I'm not running probiotic studies, and I think, as you point out, there's some companies that are doing a good job here. I would just say that a lot of the studies that are out there are ones that have a fairly low quality score or aren't even included because, as you say, they're in cell cultures or animals.
So this also gets to this point about the potential of precision medicine and what a program like Jonah offers. You could consider this is a necessary step to precision medicine, that you don't just say, one type of probiotic fits all. And one approach that companies have made is that you don't just add one strain or one species of a probiotic into a supplement, but use 10 or 20, so to cover the whole spectrum. So even though people have different baseline microbiome ecosystems, this mix of a large variety would be good for all of them in some ways.
Possibly. I do think there is a really exciting opportunity for precision medicine with the microbiome that even goes beyond probiotics. Because we know the microbiome can metabolize drugs, that we know that the microbiome interacts with the immune system. There have been several studies that have looked at, with assessing the person's microbiome state pre-treatment, you can tell efficacy or you can predict accurately the efficacy of different drugs. And that includes drugs like immuno-oncology drugs, drugs like infliximab, as well as these new GLP-1 receptor agonists that are out there. In each of these cases, we have literature that our AI has read where we can look at the person's microbiome configuration pre-treatment, and at least according to the literature, this person may or may not, or would likely do better or worse on these different drugs. And so, we're so used to precision medicine and oncology, but I think there are real opportunities with the microbiome to extend precision medicine to GI and to metabolic disease as well.
So do you intend for your company to ultimately get into this wider field of precision medicine?
100%. My goal in creating Jonah is to really use AI to understand the role of the microbiome in human health and fully understand the role of microbiome in human health, both from a diagnostic standpoint and a treatment standpoint, and then take the next step ultimately of re-engineering the microbiome from state A to state B so that you can move it in a better direction. So we're starting with this initial test where we do deep sequencing. The AI reads the literature, summarizes what's known, summarizes what you can do about it. Ultimately, our ambitions are much greater than that, to really build the foundation for understanding the microbiome and making it much more actionable. People talk about the microbiome like and say it's a new organ. But if it's a new organ, it's not like a kidney or a heart or something like that that you can see or you can study or you can take an MRI of. And I feel like that's what we really need to build is, what is the equivalent of an MRI for the microbiome? How can we build that? How can we take that data, that really complex data, and put it in a visual environment for a doctor to be able to interrogate it, to ask certain questions of it, and to really be able to answer those questions to help get to the right diagnosis and ultimately the optimal management plan for that patient.
One other, I mean, there's so many really exciting potential applications of this, and I'm sure it will revolutionize medicine sooner or later. I mean, it's just the beginning, I would say. This ability is another ability that, the microbiome holds promise for is that that you would be able to predict in a healthy person or in an at-risk person. It's just like with the risk genes that you would assess somebody in their teenage years and say, particularly somebody, for example, who has a family history of Parkinson's or Alzheimer's, and you could assess the risk of that person developing. And there's a long time span. So we know, from the time it starts in the gut, like Parkinson's, it could be 12, 14 years before the first neurological abnormalities kick in.
And as you know, oftentimes you have GI issues as an
Yeah.
initial presentation too.
Is this something that you see in the future when there's more evidence to support this and more? I mean, the longitudinal studies are hard to come by because, which investigator would want to wait 15 years, to actually confirm the hypothesis. But I think there's a growing number of studies that I would say, particularly in Parkinson's, that you could assess the risk early on and then have a long time, have 10 years of possibility of intervening in a prophylactic way. Do you see this as one area that you might get into as well?
Yeah, and this is one of the reasons why we're starting with a testing service like we are now, where the goal is to get as much value and as much, scientifically rigorous value out of a microbiome test today, and start there and use that as a starting point to be able to collect more data and to really be able to interact with patients and doctors over time to see how they're using the information, to see how their microbiome is changing, how that relates to disease processes, to interventions that they apply. And I think, to your point, and I think one of the really exciting things about the microbiome in general is that as we're able to build these sort of predictive biomarkers, the microbiome is changeable, right? I mean, if you take the teenager example that you brought up and you detect a BRCA gene mutation, okay, so now that's very useful, right? This woman is at higher risk, much higher risk for breast cancer, and you can take action on that one way or the other. But you can't change that BRCA mutation. I mean, maybe in the future you can CRISPR it up and change it, but today you can't. With the microbiome, if we do detect a signature like that, I think we will have the opportunity to re-engineer the microbiome in a way that moves it in a positive direction, so that association is no longer there. And again, we don't necessarily understand causality. We don't always understand causality in the genetics space either. But what we do know is that if we detect one of these signatures, it's at least an early predictor. And if we, to your point, if we could detect Parkinson's 10 years before the onset of neurological symptoms, that offers many opportunities to treat the patient or to work with the patient and also develop new therapeutics that ultimately can help prevent the development of that disease or slow the progression at least.
The possibilities are really unlimited, and given the speed that this field is moving forward and the number of publications that have been increasing exponentially, you think things are in the pipeline that you can't even think about right now. And at the moment, I mean, my personal assessment of this is, may not be shared by many of my colleagues, but in terms of what's come out of microbiome research in the preclinical realm, that has been, paradigm shifting. Even though it's 90% from the mouse, what's come out in terms of effectiveness in the human realm, that there's a much bigger question mark still there, because we may not have the current probiotics may not be the most effective ways of changing it. Obviously diet is a huge influence. A lot of people are not willing to change their diet. They want to take their probiotic but not change their lifestyle. So I'm a lot more skeptical at the moment, based on the current data, that this is fundamentally changing how we deal with diseases. But the potential that we just talked about is amazing. That
Yeah. And I think one of the things that I keep coming back to personally is all of the literature on fecal transplants. And, obviously, a lot of the attention has gone on recurrent C. difficile infections, and now we have the first FDA-approved therapeutics using the microbiome to treat recurrent C. diff. But, as there's so many individual case studies of someone receiving a fecal transplant and it having a really dramatic impact on their health.
Mhm.
And the thing that really strikes me, though, is that it's very hard to predict who it's going to work for, who the donor should be. Obviously, some people have had adverse events associated with these, and yet what it tells me is that if we could really understand that, and we could really understand the role of the person's microbiome in that disease process, and how we would want to engineer it in a way that would move it in the right direction, that it would really offer the opportunity potentially to cure some diseases in the way that we've seen with some people with fecal transplants. And we're a long way today from really understanding all those variables, really understanding that link between the person's microbiome and the disease process, as well as the what kind of donor, or like what we would need to change the microbiome to in order to affect it, or the ability to change it to that state. But the fact that it can happen, and that there's so many different examples of it, gives me hope that as we are able to understand these questions better, that we'll really be able to make a dramatic improvement and difference for individuals suffering from some of these diseases.
Coming back to your comment about all these different variables. So one big variable, obviously, in terms of the microbiome, is diet, and I just wonder, how does diet enter your interpretation of your sequencing data?
So we ask the patients about their diet, and so we ask a lot of questions about the kinds of food they eat. Because we're basing the analysis and the recommendations on the literature, oftentimes that literature is not, looking at the individual's diet, pre-study, right? So oftentimes they'll say like, okay, well, we took people that had this disease, or we took people who were healthy, and we put one on a meat-only diet and one on a vegetarian diet, and then we change and then we assess their microbiome at the end of 3 weeks, or whatever it is. And so there are those kinds of studies. And so we know from that body of literature that certain dietary changes can affect your microbiome in specific ways. But we don't use the person's diet in any of the associations with a disease process. And if somebody says, okay, well, I don't want to adopt a Mediterranean diet because I'm a vegetarian, okay, that's up to them, right? But all we can say is, this is what the literature says would help you potentially in your situation with your microbiome and the issues that you tell us you're having.
Okay. Yeah, I mean, this has been a fascinating conversation, I have to say. I've talked to a lot of people who are in this commercial space of testing the microbiome, and this, talking to you has certainly been one of the most exciting conversations. And on virtually all the points that you bring up. I mean, this is particularly reassuring to me. Is you are in many ways a pretty strict take a pretty strict scientific view on this, and not trying to sell products in the background. So I think this is really an exciting, and I can just see in the future these various opportunities that we touched upon evolving as major applications of your approach.
And I think to your point, trust is so important here. I think we all saw the criminal blow up with Ubiome a few years ago. Beyond that, you see people in the supplement world or in other parts of the microbiome world make wild claims that are not always supported. And yet, to me, you look at the science and there's real exciting stuff there. And so it's very important for us that we be very transparent, and that we earn the trust of doctors and patients, because in particular because of some of this other noise out there, and really always be driven by that North Star of providing the best information, but only what we can say and not beyond what we can say.
Yeah, I mean, unfortunately, this, what you just called noise is actually a pretty strong, background noise that often obliterates the signal, that should come out of this. And this is several reasons. I mean, people now, authority is no longer academic credentials, but it's number of Instagram followers. Very good examples, that you see on the internet that I would say it's the main criteria that the majority of people actually follow. They don't. On the other hand, scientists have not taken on the task of taking this themselves to the public. There's a reluctance of scientists to sort of spend any of their time not writing NIH grants or doing the research, but actually communicating with the public, which I think is equally important.
Well, at the same time, if when I go to DDW, I see, I counted, I think, 450 talks and posters on the microbiome at the most recent DDW. And the GI docs that I talked to, the pharma industry, they all know that there's something there. And I would actually say that I've yet to meet a doctor that says the microbiome is not important. I think that there's a difference in opinion on when we'll know enough to have clinical translation. Some people think it's around the corner. Some people think it's 10 or 20 years out. But I've yet to meet somebody who doesn't feel that the microbiome is really important, and that if we understand it, that we can really level up on our diagnosis and treatment of a variety of diseases. And you look at people like Jim Allison at MD Anderson, who invented immuno-oncology, won the Nobel Prize. The gut microbiome is a key area of their program for really understanding how the microbiome impacts, plugs into the immune system, and how that affects cancer treatment. And so really some of the world's top scientists are working on this. But he's not out on Instagram or running ads. And so I think, to me, the truth of the science and like the excitement of what's there is really what's important. And I think if we are representing it effectively, that ultimately will win out.
Yeah, I think this is a good point to close our conversation. It was really exciting talking to you. I have to say, looking forward to collaborations in the future with your programs. Sounds very exciting. And yeah, I mean, if you still want to say any final statement from your side.
No, this was really wonderful. I really appreciate the work that you do. I'm excited about your PBS special, and I really appreciate the work that you've done to elucidate and illuminate the role of the gut-brain axis and that mind-gut connection. Because even though a lot of GI doctors are paying attention to this, I see less of it in the neurodegenerative world. And I think there's an enormous opportunity here. So I really appreciate your work.
Yeah, so thanks, Leo, again for the time you've taken to do this conversation. And yeah, as I said, look forward to interacting with you in the future.
Thank you so much.